Hello, everyone, and welcome to today's webinar, IOTest Myeloid Activation CD169-PE HLA-DR-APC CD64-PB Antibody Cocktail, Visualizing Immune Response During Infections. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckman.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Shankar Pattabhiraman, Dr. Shankar Pattabhiraman, Technical Product Manager for the Flow Cytometry Business Unit with Beckman Coulter Life Sciences. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Shankar, you may now begin your presentation.
Hello, ladies and gentlemen. My name is Shankar Pattabhiraman, and I am a Technical Product Manager with the Global Flow Cytometry Business Unit at Beckman Coulter Life Sciences. Today, I would like to talk to you about our new assay, IOTest Myeloid Activation, and how we can use this to visualize the immune response during infections. Next is a standard disclaimer for intellectual properties. Bacterial versus viral, to treat or not to treat is the question. Why is this so important? Differentiating between the natures of infection can allow healthcare providers to deliver quicker and more accurate treatments. Antibiotics are effective only against bacteria and are not useful in the cases of viral infections such as colds, flu, and more sore throats. Due to the lack of rapid and good tests to differentiate bacterial infections, there is a tendency to prescribe antibiotics erroneously in the case of viral infections or excessively. Antibiotics have saved millions of lives and have contributed to a significant increase in the expectancy and the quality of life. But the overuse or the misuse of antibiotics threatens to create a global problem of antibiotic-resistant bacterial pathogens. This is a significant threat to the public health system and comes at great cost in terms of life and money. The main challenge in the effective discrimination between bacterial and viral infections is that the clinical features presented, such as fever, cold, pain, are generic to both of them. Microbiological tests, that is the culture of bacteria in vitro, are dependent upon the accessibility to the infection site, sampling efficiency, and the time to result can be long, 1 to 3 days, which makes treatment problematic. There are many molecular tests available for the diagnosis of viral and bacterial infections. Many are even point of care. However, these are available only for a small set of infections. Further, prior knowledge of the microorganism is required to develop a new molecular test, like the sequence information, which makes new pathogens difficult to diagnose. Current protein and immunological tests like CRP, procalcitonin, and cytokines have low sensitivity and specificity. Thus, there is a need for an effective, rapid, and easy screening test. Infectious diseases are caused by the invasion of the body by microorganisms. Upon infection, these are rapidly recognized and eliminated by the innate immune system, which is the first line of defense against pathogens and reacts within hours. Myeloid cells, such as dendritic cells, monocytes, and macrophages and neutrophils, are activated upon contact with the foreign bodies and microbial patterns. Their recognition by the innate immune cells leads to the initiation of a robust inflammatory response, including the release of various pro-inflammatory mediators such as cytokines and interferons, and the recruitment and the activation of other myeloid cells. The innate immune cells provide an early, however non-specific response to infections, taking place within the first 12 to 24 hours of the infection. The myeloid cells then activate the slower but more specific and effective cells of the adaptive immune system. The adaptive immunity, which is required for the long-term sustained response to infection, is activated through the classical MHC TCR interaction with the activated dendritic cells. Interferons are cytokines secreted by a variety of cells upon infection, inflammation, and autoimmune diseases and have immunomodulatory effects. Interferons are at the crossroads of the two systems as they are essential not only for the innate immunity but also for the activation and sustenance of the adaptive immunity. During bacterial infections, it is predominantly the type 2 interferon, interferon gamma, which is secreted by the NK cells upon activation by the dendritic cell IL-12. Viral infections, on the other hand, lead to the production specifically of type 1 interferons such as interferon alphas and betas. Thus, interferon signaling is also at the crossroad in the innate immune response as it is able to differentiate between infections by bacteria and viruses. However, direct measurement of interferons is a challenge. The short-lived nature of interferons make direct measurements difficult. However, interferon signaling pathways can be observed by the downstream genes and the markers activated by these molecules, sort of an immune imprint of interferon signaling. Several cell surface markers are specifically activated on myeloid cells upon the secretion of interferons. Ideal markers of interferon response to infection would be specific to viruses and bacteria, helping to differentiate between the two. This is the case for monocyte CD169 and neutrophil CD64. Interferon type 1, interferon alpha and interferon beta specifically upregulate the expression of CD169 on monocytes and is specific for viral infections. Type 2 interferons, that is interferon gamma, specifically upregulates the expression of CD64 on neutrophils, which is specific for bacterial infections. The specific differential markers of infection and interferon signaling are CD169 and CD64. We have also included HLA-DR as an indicator of the immune status to the assay platform. CD64 or Fc gamma receptor 1 is constitutively present on macrophages, monocytes, eosinophils, and only to a small extent on resting neutrophils. However, neutrophil CD64 expression rapidly increases in the presence of bacterial cell wall components and some pro-inflammatory cytokines such as interferon gamma and granulocyte colony stimulating factor GCSF. Neutrophil CD64 expression have been reported in many bacterial diseases showing it to be a potentially useful early biomarker for detection of bacterial infection and sepsis with high sensitivity and specificity. The CD169 cyclic 1 gene expression is induced by type 1 interferons on myeloid cells such as monocytes and dendritic cells. Recently, CD169 has been identified as an inducible viral attachment factor on myeloid dendritic cells. CD169 is not constitutively expressed, and the induction on monocytes of this marker has been shown in a number of different viral infections such as HIV, Epstein-Barr virus, and with type 1 interferon expression. Monocytic CD169 has been shown to be a biomarker of HIV infection and disease progression. Further, it was shown that antiretroviral therapy led to a decrease in the monocytic CD169 corresponding to the decrease in the viral load. Monocytic HLA-DR, an activation marker, is upregulated in acute infections, both viral and bacterial infections. The levels of HLA-DR, however, comes down as the infection progresses due to immune. HLA-DR in conjunction with the specific infection markers CD64 and CD169 provides a snapshot, valuable insight into the timeline of infection immune response. Further, decreased expression of HLA-DR on monocytes is one of the best studied markers of sepsis-induced immune suppression. Together, monocytic HLA-DR and neutrophil CD64 form a potent combination of biomarkers for an accurate assessment of immune response during septic shock. With all this information, a three-color panel has been put together called the IOTest Myeloid Activation with CD169-PE, CD64 specific blue, and HLA-DR-APC for visualizing the immune response during infection. The flow cytometric assay can be used to visualize the different aspects of innate immune response during infections. It is a ready-to-use pre-mixed and titrated cocktail. It's rapid and easy to use. Just add blood, the antibody cocktail, and VersaFix lysis solution to the tube and vortex. The sample is ready for acquisition in 10 minutes. There are no washing or centrifugation steps. As you can see, the fluorochromes are on 3 different lasers, which means no compensation is required, which makes the analysis really easy. Our R&D team has collaborated with a number of international groups to generate publications which show the utility of these markers CD169, CD64, and HLA-DR in visualizing the interferon and immune response during infections. Our data demonstrates that the upregulation of these markers is very specific to the type of interferons used, as seen from the figures. In vitro experiments showed that type 1 interferon specifically induced the expression of CD169 on monocytes and not interferon gamma, which is the type 2 interferon. Similarly, interferon type 2, that is interferon gamma, upregulates neutrophil CD64 expression specifically and not any of the other interferon alphas or betas. The two markers are thus able to differentiate the interferon response in a rapid flow cytometric test. Further, when these markers were used in the analysis of blood from patients presenting with fever in the hospital emergency department, we observed that monocytic CD169 and neutrophil CD64 were effective biomarkers for the differential infection immune response. As you can see in the flow cytometry plots, there is an overexpression of CD169 on monocytes in the case of viral infection, while bacterial infections led to an overexpression of neutrophil CD64. Analysis of a cohort of 138 patients entering the emergency department of the La Hospital, Marseille, France, showed that the quantitation of the two biomarkers by flow cytometry, the MFI value, as shown in the y-axis, was effective in differentiating bacterial and viral infections, as can be seen in the plots. The cohorts had a wide range of bacterial and viral diseases. Thus, these markers can be used as generic markers of bacterial and viral infections. Several other cell surface biomarkers were also tested in this patient cohort. Neutrophil CD64 was observed to distinguish wide variety of bacterial infections with high accuracy. Monocytic CD169 was not a significant marker for bacterial infections as was expected. With the IOTest Myeloid Activation, we have a rapid flow cytometric test for visualizing the differential immune response to bacterial and viral infections. The panel can be used in multiple research areas such as interferon signaling, antibiotic resistance, viral disease progression, and many others. It can be an effective assay to study the innate immune response and interferon signaling during infections, inflammatory diseases. The kinetics of interferon release and characterization of the different interferon classes in various disease states can be performed easily, which is useful not only in the understanding of the innate immune response, but also the activation of the adaptive response. Rapid identification of bacterial infection is essential in understanding and combating antibiotic resistance. Further, the panel can be used in the development of biomarkers and new antibiotics. Type 1 interferons play an important role in viral infection and progression. Markers such as CD169 is very effective for studying the dynamics of interferon response during viral infection. We are in the middle of a COVID pandemic. Number of publications have shown the effectiveness of interferon signaling as a biomarker of SARS-CoV-2 infection and COVID disease. Several transcriptomics analyses have shown interferon signaling and the induction of expression of interferon-stimulated genes, ISGs, as an important signature of SARS-CoV-2 infection. Like these examples from the analysis of myeloid cells in the BAL fluid of COVID patients, showing the expression of numerous interferon signaling genes is at the top of the list. The study also looked at the expression of interferon 1 response, ISGs, and the direct measurement of interferon levels in COVID patients who were stratified according to the severity of the disease. There is an increase in interferon type 1 response upon infection. However, interestingly, the interferon response significantly decreases with the progression of the disease to more severe manifestations. Thus, monocytic CD169 can be an effective biomarker for SARS-CoV-2 infection and for following disease progression in COVID, as it's a very good marker for type 1 interferon. So how does the myeloid activation assay work with SARS-CoV-2 infection? Studies performed by our collaborators in the CHU Montpellier, France, on 53 patients in the COVID emergency unit showed that monocytic CD169 by flow cytometry was highly correlated with COVID positivity with qPCR. This rapid flow cytometric assay can be effectively used to identify COVID positive cases who can be confirmed with PCR. This study was recently published in the Journal of Infectious Diseases. Another study in the CHU Marseille, France, with 155 patients confirmed these results. In another publication, the flow cytometric assessment of different monocytic markers in a large cohort of COVID cases showed very good correlation of CD169 and HLA-DR on monocytes with disease onset and progression. As seen earlier, there is an increase in monocytic CD169 with infection, which drops down as the disease progresses or with disease clearance. With the IOTest Myeloid Activation, we have a rapid, easy to use test for visualizing the early stages of infection, a snapshot of the innate immune response. We can effectively differentiate between the bacterial and viral infection immune response and have a good idea of the interferon signaling during infections. The test is easy to use, a single step, no wash, no compensation, and a rapid assay. The data and publications were generated with our collaborations with key cooperation partners in France and Germany, in the hospitals in Marseille, Lyon, Montpellier, and in Berlin, Germany. Here is the ordering information for the kit. Thank you for your attention, and I am ready for your questions. Thank you very much.
Thank you, Shankar, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Our first question is, what do you think is the real advantage or breakthrough with this assay? Shankar, can you hear me? Okay. Shankar, I'm going to ask you a question and then I want you to answer it, if you can hear me. What do you think the real advantage or breakthrough with this assay?
Okay, so
Okay, Shankar, I'm going to ask the question again. What do you think is the real advantage or breakthrough with this assay? Can you hear me? Shankar, can you hear me?
yeah, I can hear you now. Yeah.
Okay.
Sorry. There's some problem. Yeah.
Sorry. Shall I read the question again?
Yes, please. Thank you. Yes.
Okay. What do you think is the real advantage or breakthrough with this assay?
Hi. So I think the real breakthrough, the real importance of this assay is that there are not many assays available to study the activated myeloid cells or even the innate immunity during infection. So this is sort of a black box, and even if you have assays, they're complicated to do. So with a simple flow cytometry test, we can quickly have an idea of what's happening to the immune system during infection. That's number one. Second is again for interferon signaling, it's pretty difficult to actually have an idea of what's happening, to what are the inflammatory processes during infection and other during infection. And this is again a quick and easy assay to look at or have a snapshot of what's happening there. And I think these are the two main points I think I would like to emphasize with this assay, that we're able to have an idea of a glimpse of what's happening in the innate immune. Because most tests are usually with the adaptive immune system, the T cell, B cell response, the antibody response. But this is a little bit difficult to actually have an idea about.
Okay, thank you so much. I've got another question for you.
Mhm.
Can this test be used to study other inflammatory diseases such as autoimmune diseases?
Yes, there are. So I've spoken mainly about I've spoken mainly about infection response and all those things, but this can be used to study the innate immune system in a wide variety of conditions. And the best studied the conditions are, of course, the autoimmune diseases, the inflammation during autoimmune disease. So recently, there's been a publication which has shown that CD169 can be a very good marker for systemic lupus erythematosus, and also in Sjogren's disease. And even in systemic lupus erythematosus, they show that there's an increase in the CD169 levels along with the disease progression, and can be used as a good marker for disease progression. And even when the patients are treated with hydroxychloroquine, you can observe a reduction in CD169 corresponding with clinical improvement. So it's actually a very good marker or a good way of monitoring autoimmune diseases and all inflammation associated with these kind of conditions. For example, yeah.
Okay, great. Thank you so much. I have another question. What happens when you have both viral and bacterial infection at the same time?
Yeah. So what you would actually see is both of them will get upregulated. Both CD169 and CD64 will be upregulated when you have both the infections there. In, and it and since the specific since both the markers are very specific, so you can actually be sure that both infections are present. No, it's not a non-specific upregulation of a marker during the infection. Yeah.
What about testing children and infants? Where other testing methods might be difficult to use?
Yes. So this is absolutely I think one of the big advantages, because it's a blood test. So it has got definite advantages over other ways of sampling, like like a swab or different, to get access to the infection site, or it's pretty complicated to do. So a simple blood test with a flow cytometric assay after that actually makes it very easy to test, especially children and infants, because all you need is a drop of blood. The assay just requires 25 microliters of blood. So this is particularly, I think, will be useful when we are doing research or looking at cases like that.
Okay. Next question. Is this a diagnostic test?
No, this is a research use only test and should not be used for any diagnostic purpose.
What kind of research topics can you envisage for COVID research?
Yes. So as I've shown that people have observed interferon signaling as a pretty good marker for COVID research. That is at least to look at disease infection and disease progression, and even disease severity and clearance. So this could be a test, I think, which can be used for studying the role of different cell types, studying the role of different treatment schedules, what's happening to the COVID symptoms, And I think this is because the current test is actually what you call it's a PCR test. So you're measuring only whether there's presence or absence of the virus, but you're not seeing the reaction of the body to it. And most of the other tests which are done for doing that are basically physiological parameters. So here with this test, you are able to look at the what is happening to the immune system and how the drugs, vaccination, and everything is going to affect the immune system and help it fight the infection.
Do you think there are applications for this test in the present pandemic?
Yes, I would still think that though PCR is the gold standard test and the perfect test for diagnosis, there is a lot of there is scope to use this test in monitoring patients to look at how the patients are progressing, especially in hospital settings. So this could be a marker of seeing whether the disease is progressing, it's becoming severe, the patient needs more radical treatment, and also the effectiveness of the vaccination process. So I think this can still be used to measure those kind of aspects in a research or in a clinical research setting.
Okay, and other than people walking into the doctor's office with a fever, what other settings do you think this assay can be applied to?
Uh, yeah, I mean, as I said, so fever is one of the questions where they can do, but you can think of mass screening of people. So you can also look at places where you are, like in workplaces or other public places where you are thinking of actually seeing whether the patient is currently having a infection, and you want that not to spread, or the kind of quarantine measures. And this can be a quick way of also checking whether the patient is infected or not, to have an idea at least before a confirmatory PCR is done to be sure that is happening. So,
And how do you overcome possible non-specific staining?
That's a good question. So we have actually from the assays we have done, and from the types. So first of all, the antibodies are titrated. So we have actually a very specific concentration of the antibody into it. So that also is reducing the non-specificity. And the second thing is CD169, there is no expression in normal cells. So it's when you see an increase in expression of CD169, you can be sure that it is an infection, it's a viral infection. You can be pretty sure of that. CD64, yes, you do have a little bit of staining with CD64, even in resting cells, especially in neutrophils. So, I mean, because monocytic CD64, it is constitutively expressed. So here we are talking about neutrophil CD64. You do have a slight amount of expression, but the upon bacterial infection, you have a very clear overexpression. So I don't. So far in different clinical settings, we don't see a problem of background staining.
Okay, next question. Simple techniques like PCR or any such assays can also detect the expression of genes like HLA. What is the advantage of this assay in case of expression analysis? Is it in the perspective or sensitivity and specificity?
So I think the biggest difference is so molecular tests are very good because they are very you can see what the expression of the particular gene is. You can very accurately measure that, but you really do not know what the physiological impact of it is. So you can have an expression of a particular cytokine or the cytokine gene, or overexpression of an interferon gene in it. But to know what the physiological impact is, what's happening physiologically, what's happening to the immune cells because of the overexpression of it, you need a cellular based assay. So that's the advantage of this assay, because it gives you a cellular idea. It tells you that, okay, HLA-DR, there's an increase. That means you see that the monocytes have been activated, and that's why you have an overexpression of HLA-DR. Same thing in sepsis shock condition. You see a decrease in HLA-DR because you have an effect of the sepsis inflammation on it. This might not directly correspond with the gene expression of HLA-DR in the cell, but this is the physiological or the cellular condition in which it's there. So I think that's the advantage of having a flow cytometry test where we are measuring the physiological impact.
Okay, next question. What is the positive predictive value for COVID-19 of this test?
Yeah. So again, let me be very clear. This is a research use only kit. So we cannot use it for diagnosis of COVID, and it's not a specific test. So you understand this is for all viral infections. But this can actually help you in screening, or it can help you in actually looking at patients. So you can actually do a screen to see how many of them are positive, and you can do a further confirmation with PCR. So this is the advantage of this. And as you can see from the publications where in the clinical research setting, they have looked at qPCR data and the CD169 expression, they see a very, very high correlation between the two. So the publications are there. So you can actually go to the publications and read. I think the specificity and everything is even mentioned. So you have the data in detail out there.
And can you use a finger stick for the blood sample?
Actually, yes, you can use a finger prick for the for the blood sample. So again, since it's a research use only kit, so you need to standardize your methods. We cannot tell you that this is exactly the way to do it. So you need to do a little bit of standardization. But as you can see from the publication, so during the development of the assay, we did study finger prick blood. So like with a glance, you take a drop of blood and the assay you can perform with it, and you have very clear demarcation of the three cell populations, and it can be very easily used with a finger prick blood also. But it needs to be standardized in your lab.
Okay, thank you so much. Next question. Can this assay be used to understand the role of tumor microenvironment to identify the related biomarkers?
It is. I actually have to think about it. So it is a little bit complicated in this because we are looking at circulatory cells. So there's mainly cells which are actually circulatory in cells in the blood. So to measure what's happening in the tumor biomarker would be difficult. But that said, if we can access the tumor site, so suppose you're doing you are having so if you can access the tumor site, you can get a clinical sample, then you can actually measure you can actually see the activated myeloid cells in that place. But you will have to access the site. You cannot use blood then.
Okay, Shankar, we have time for one more question.
Oh,
how can we set up the cutoff for the CD64 neutrophils overexpression, and if there is a link between the severity of infection and the degree of that overexpression?
Okay, this is a very good question. Actually, it's so the only way to set up the CD64 neutrophil expression for bacterial infection is to follow the infection. And so basically you need a gold standard test that is either what you call antibiotics, so either the clinical follow-up of the patient or a microbiological test to confirm that it's a bacterial infection. And with that, you can actually set up the CD64 expression. I'm not too sure whether we can directly quantify the severity of the infection with the neutrophil CD64 expression. I don't think these two markers are directly linked to one another, but I think you need more data for it. definitely. But so far, I don't think it exists out there.
Well, thank you again, Shankar, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.